Thank you, Alke. As you can see also from the program, I also in charge to make time because we were going against time because we started 11:50. Yes. So that's fantastic. So could I kindly ask to Professor Bard to come over? He is, since 1993, is a professor in cellular biology and then become a founder of a company, which is a good path from the nanomedicine perspective. And his specialty is in scavenger cells, if I believe correctly. So the title of his talk is Role of Specialized Hepatic Scavenger Cells in Control of Biodistribution and Biologics and Nanopharmaceuticals. So you have...
So thank you for the introduction. I'm here to talk about special liver cells that are engaged in uptake of blood-borne macromolecules and nanoparticles. As you can see, as you already heard, I represent my university lab, and I also started a company, and we are doing things in parallel, but we have a firewall between us. So, okay. I'm going to start by showing you this animation, which is a highly simplified, but it illustrates nicely the point I'm going to talk about. Not whenever, but very often, when macromolecules and or nanoparticles are injected intravenously, the result is a very rapid uptake in liver. This is a fact that we have to live with, and it's a pain in the back for people who develop new and promising drugs. What is it that we see? This is the focus of my study. What is going on in the liver in detail? This is also a type of information that should be of great interest to people who don't like this uncontrolled uptake in liver going on. All right, so what do we see here? Okay, we see a table showing the distribution of liver cells. If you talk about liver in terms of volume, the liver is a bag of hepatocytes, the big liver cells. And therefore, there is a tendency to think about the liver as a bag of hepatocytes. Everything that's going on in the liver is therefore the result of hepatocyte activity. This is very wrong when it comes to understanding of how the liver deals with circulating macromolecules and nanoparticles. First of all, because when you talk about the cell number, the hepatocytes or parenchymal cells make up only 65%, and you have then the sinusoidal cells making up quite a high number of cells. That's one thing. And the other thing we should be aware of is the fact that these liver sinusoidal cells make up the wall of these fine liver capillaries or sinusoids. And all blood that enters liver will pass through these sinusoids. And then these sinusoids are lined by endothelium. And this is a very special type of endothelium. You also find liver macrophages or Kupffer cells. I'm going to focus on these endothelial cells. Unfortunately, we have only one name for these cells, and that's endothelial cells. They behave very differently than conventional endothelial cells, to put it this way. If we enlarge the sinusoid like this, we get a half pipe of this sinusoid, and you can see that well, the artist has put some receptors on the surface of the endothelial cells. And whenever a nanoparticle or macromolecule enters not whenever, but very often, these receptors will recognize these molecules and engulf them, take them up, and metabolize them if the cells have enzymes to metabolize them. So this is another cartoon that our artist made, trying to highlight what's going on. This is, again, very simplified. But what you should pay attention to here is, of course, the yellow little small explosions going on here, meaning that the molecules or nanoparticles are taken into the cells and metabolized. And you should also look at the receptors. They have a very, very high cycling time. They're not sitting there as fixed receptors. They're constantly bringing things into the cell and then popping out again. It's a matter of seconds. And these cells are extremely efficient in uptake of circulating nanoparticles and a whole bunch of macromolecules that should not be in the circulation. Okay, that's the ad for a little company. So what sort of macromolecules are, or materials are recognized by these receptors and cells? Well, all kinds of biomolecules are recognized. So that's the simple answer. I won't dwell with this anymore. The focus, again, of my talk is the liver sinusoidal endothelial cell. And the major receptors expressed by these cells are depicted here. They also have other types of receptors, but these are the workhorse receptors. They're very efficient, and they are uniquely expressed on these cells. It might surprise you, but the mannose receptor is expressed only on these cells in liver, not on the liver macrophages. This is okay. And the major scavenger receptor is called the stabilin 2 or stabilin 1. It's also uniquely expressed on liver sinusoidal endothelial cells. And then you have the special Fc gamma receptor 2b2, which is also uniquely expressed on these cells. These receptors are responsible for endocytosis, receptor-mediated endocytosis, not phagocytosis. Only these cells cannot perform phagocytosis. They are specialized on receptor-mediated or clathrin-mediated endocytosis. A few more words about the endocytosis receptors. Again, you have the mannose receptor. There are many, many ligands that are taken up, both externally or foreign molecules and own molecules. The same is true with the stabilin 2 receptor. Yesterday, there was some talk about hyaluronan as a stealth molecule. It is not a stealth molecule. If you want to target the liver, put hyaluronan on something. This is the way we discovered this receptor. It is a very high activity, high affinity receptor for hyaluronan. All right, and the Fc gamma receptor is also a special type of receptor that recognizes soluble colloidal immune complexes formed with IgG. And you don't find this receptor in other liver cells. All right, so we're now talking about a so-called dual cell principle of waste clearance in liver. And this is important if we ever should have a hope to understand what's going on in the liver in terms of uptake of nanoparticles or macromolecules. We have 2 points to think about here. One thing is that we always have to distinguish between phagocytosis and pinocytosis. This is textbook material. It's okay. This is textbook material and nothing special about that. Then what is very important also is that blood clearance in vertebrates is performed by 2 distinct cell types, the macrophages And they are professional phagocytes. They will take up large particles. And you have the scavenger endothelial cell or the liver sinusoidal endothelial cell, which is the professional pinocyte. And they do this division of work between them. So this is next to my last slide. Size matters. You have the liver sinusoidal endothelial cells here, and you have the Cooper cells here along this size scale. And and the particles and macromolecules will distribute in the cells about the way they are shown here. So the conclusions is that we need more detailed knowledge about this so-called dual cell principle. We need it in order to design strategies to control liver uptake of biologics and nanopharmaceuticals, and we need it to understand the mechanism of hepatotoxicity caused by administration of biologics and nanoparticles. So with this, I thank you very much.
We have, for the sake of time, we have time for one question. Yes?
Actually, it was super interesting topic for me, at least, because I have been working on liver stellate cell targeting and using mannose-6-phosphate receptor targeting and PDGF receptor targeting. And one of the things which came to my mind is, of course, that was new for me, that mannose receptor is not expressed on macrophages or Kupffer cells, but on sinusoidal. Although the notion is that it's present specifically on Kupffer cells, they are like M2, like the macrophages. What happens to the sinusoid during inflammation and fibrosis?
These cells will gradually disappear, and if the fibrosis is allowed to proceed, and the scavenger function performed by these cells will gradually decrease, and in the end disappear, and that's the end of liver, more or less.
Thank you.
I would like to thank Professors